And I have here both of the beakers that were run at the same time. This was the one, this was sort of the control, just the cell that I ran in distilled water, right? And this was the cell that was set up to run in the iodine that you can see the time-lapse video for. And there's a pretty big difference in color, right? I'll move away so you can see that. The cell that we stuck in that iodine water, over time, it turned this funky black color, right? So the question then is, A, why did this turn black? What's going on? Note also that the water in the beaker stayed that golden yellow iodine color. It didn't change to black. The black is only in the cell. So I want you to remind yourself what component that's in the cell could have turned colors, because maybe this stuff in the beaker, maybe this yellow stuff, the iodine, interacted with it. Maybe there was some movement. In this case, it wasn't from the cell to the beaker, but maybe something in the beaker got into the cell and caused a reaction. What could that have been? Okay, so I'm back. It's been an hour. Here is the cell we made earlier that's been soaking in distilled water. See it right here. Looks pretty much exactly the same as when I first made it, right? But what we want to do now is we are going to weigh it again, right? See if I can cover up somehow I magically covered this up last time. We're going to weigh it again, see if maybe it's gained or lost any weight, right? So I'm going to take it out. Put it over here. And now the weight is 28.32. So you want to write that down. That's the second weight. I'm going to go get my lab manual and put it down. 0.33 grams. Okay. There's a reason for keeping track of the weight. All right. The other thing we're going to do now is I'm going to carry out a couple of tests, because one of the points of this lab is to figure out if the stuff in the cell meal, the stuff that is in here, the glucose, the starch, the protein, does that stuff, and the water, can that stuff leave the cell, or does it just stay put? Like, can it leave? Can it travel into the water in the beaker, or does it just stay put in the cell? So we need to run some tests to see, did the glucose move from here to the water in the beaker? Did the protein move? Did the starch move? Did the water move? I can test for pretty easily for, and you've actually tested for one of them already. You've already tested now for, did the water move? You have an answer, you just may not know it yet. The other two things I can test for using this tube and this beaker are for glucose and for the protein. So I'm going to explain to you what I'm going to do. We already know. We know that there was glucose and protein in here because that's where we put the cell meal, and it was in the cell meal to begin with. So really what we want to know is, did the glucose and the protein travel into the liquid in the beaker? So we can test the liquid in the beaker to see if it contains those things. What tests could we run to find out if glucose or protein is present in the beaker water? Well, you may be familiar with these back when we did the biomolecules test, right? We have Benedict's solution, which is for simple sugars, and Burette's solution or reagent for proteins. So I'm going to set up some test tubes. I'm going to set up a negative control for each one, and then I'm going to set up a tube that contains the water in the beaker for the other test, okay? I'm going to point the camera down so you can see what I'm doing. And I am going to go back in my lab manual to the biomolecules lab and remind myself for simple sugars, which was on page 22, how to do the simple sugars test. So, like I said, I'm going to give myself 2 test tubes here, these 2, and this is going to be for the simple sugars. I'm going to do one that is the beaker. I'm just going to put a B on it, and I'm going to put one as my negative control. So I've got one with a B and one with a minus sign. And the way to set the test of is it says put 10 drops of Benedict's reagent into each tube. Alrighty then. Right? Looks like that. In the one that is the negative control, I'm going to add 10 drops of water because that's it shouldn't have any glucose in it if it's water, right? So that's a good negative control. 1, 2. 10 drops of that. And then I'm going to put 10 drops of the solution from the beaker here that my cell's been sitting in. Put that in the other tube. And first we want to just look at the color of the tubes before, because remember what we're going to have to do to these tubes is stick them into really hot water and boil them, almost boil them, to get the results. The other test, and so hold on to those because I'm waiting for my water to heat up. I don't know if we're quite there. Not quite. The other test I'm going to set up is the Buret's test. So I'm going to flip back over to page 26, which tells me I need to put 5 drops of the, again, I'm going to do, this is going to be my negative control. This is going to be my beaker. It says into each one, first I'm going to put 5 drops of water. And then I'm going to put 5 drops from the beaker in. And then I'm supposed to put in, and this, so right now this is for the Buret's test. This looks pretty clear, right? Now I'm going to put in 5 drops of this reagent. Okay, and then for this test, you have to remind yourself when it's negative, like this one over here, that's my negative, right? It just stays that blue color. And in this case, both of these tubes look pretty much exactly the same. If you want, I will put a white background behind it too so you can see. Whether I'm looking at my negative tube or the beaker, they both look pretty blue. So I would say, since it didn't turn purple, protein, that's what this one tested for, did not go from my cell into the beaker water. So protein didn't move. Okay, so now we're going to take, these are the Benedict's test tubes, the beaker, the negative control. Here's our hot water. Let's remember they start out blue. And in case you're trying to remind yourself, what are we looking for? And I know for some reason you can't quite see these super well unless I hold my hand back behind. We're looking for a color change in the tubes, because if it changes in color to green or yellow or orange or red or even brown, then that means it's positive for simple sugars in that. Simple sugars in the beaker water, that is. If it doesn't change color, then we know that simple sugars didn't move from the cell into the beaker water. We're going to keep those in there for a minute. Hopefully they turn out the way I would expect. I'm watching real closely. I'm actually starting to see a change now. I don't know. I'm starting to realize I filmed these in the beaker from a different camera last time, but I'll make it work, right? Can you see that one of the beakers is no longer exactly the same color anymore? One's blue, one's almost a little blue-gray. It's starting to turn green. And I can actually tell that it's turning a little bit oddly orange. So, in case you're trying to keep track of which tube is which, this is our negative control here, the bluer one, and the B is from the beaker. So B is starting to turn green. The negative control is staying blue. So already we're getting a pretty good idea that the water in this beaker now contains simple sugars. And the only place those simple sugars could have come from was inside of this cell, right? So just to review, and it is even getting darker over here now. If I hold these up, you can now see for sure. This is my negative control on this side. It's still blue. My beaker water on this side started to turn that green color, kind of ugly green. So that tells me beaker has glucose in it now, which it didn't have before. Before, this water to start was just distilled pure water. So that tells me sugar moved to beaker. And if you need a reminder, the results from the burette test for protein were that no protein moved. So both the negative control and the beaker water were negative for protein. So protein just stayed in our little cell. And this is even getting darker as I go. I don't know if you can see that, but now it's even more green. So there you go.